Hello friends, I am Dr. Sutar from Sanjini College of Pharmaceutical Education and Research. Today we are going to see a new hormone that is used as a plant growth regulator. Till now we see that what are the plant growth regulators, how they are to be shown, their functions, what are the difference between the other chemical components and the plant growth regulator. In short, just we see revise this one. So plant growth regulators, or that is also known as a plant hormone, or they are also known as a phytohormone, because these are the chemical which are to be synthesized in the plant body, and they are going to control on the specific activity. Just like insulin is there in human being, insulin is tries to control or maintain the blood sugar level in the human being. Other chemicals are also there, but this insulin is responsible for that one function only. So likewise, in case of the plant body, there are the different hormones are there. They are going to show a specific activity. They are going to control this one activity in the plant body. So they are known as a plant growth regulator. They are two type of the plant growth regulators are there: endogenous, or it is also known as a native one, that is to be synthesized in the plant body, and another one synthesized synthetic one, that is to be synthesized by the chemical reaction outside of the plant body, and they are going to be supplied to the plant body. So likewise, the plant growth regulators are there that are show their function. So auxin, gibberellin, and cytosins are the growth promoters are there, whereas abscisic acid and ethylene, they are the inhibitors are there. Now let's we see the next one, plant growth regulator, that is a gibberellin. Gibberellin is the one chemical which is to be synthesized in the plant body. They are the found in case of the endogenous part of the plant body. So they are known as a endogenous plant hormone. They are to be found in the various part of the plant body, such as the young shoots are there, embryos are there, germinating seeds are there, then roots are there. In the tip portion, they are to be found. As these chemicals are there that are to be synthesized, and they are to be characterized. Later on, the names are there that are to be given. As the GA1 is there, GA2 is there, GA3 is there. Likewise, the n number of the components are there that are to be synthesized and they are to be identified. They are known as gibberellin. Why the no name is so given gibberellin? That chemical name is there that should be found. One experiment is there should be there. In case of this one, field in the field of the rice, there is a some of the plants are there, or we can say that the these crops are there that are having the somewhat heighted one. This is the one. These plants are there that is to be heighted one, whereas these are the having a small height is there. Where this one heighted, where the height is there is to be high, they are showing somewhat yellowish color, or there is a somewhat fungus infections are there that should be there. So the farmer are there that brings this one particular crop to the research lab. So there is a one scientist, he studied on this one, why this is the difference is there. So the chemical test and other analysis is carried out in both the plant, those who are having a short height and those who are having a large height along with the fungal infection. They came to know that the there is a fungal infections are there. The plant which is having a yellowish color and the plant which is having a green color or short height, there is no any type of the fungal infection is there. But those plant who are having a large height, they show the fungal infection, and the later on the various analysis is carried out and they find out that the name of this one chemical is there, that is a gibberic acid is there. So this is responsible for the growth of this one particular plant in the, or which is to be observed in case of the rice. So discovery is there. The first discovery that is from the fungal infection, that is a fungus Gibrella physicoi, and all the experiments are there that is performed by a Japanese scientist Kurosawa. he performed an experiment on the rice and concluded that the chemical is nothing but a gibberellic acid is there. And by the name fungus Giberella, they he gives the name to that one chemical, that is a gibberellin. So this is the history behind that one particular hormone, that is a gibberellin is there. So this gibberellin is utilized for the growth of the plant in a vertical directions. So what are the applications of this one? Where we are going to use this one gibberellin. So let's see. First one, it stimulate the internode elongation. Those plants who are having a short height. So in this plant, these chemicals are utilized to induce the height in this one particular plant. So how they are to be induced the height? So it causes the internode elongation, the distance between the two node. Node means what? Where the leaves are there that are to be originated. So the portion is there is known as a node. Node is a region, and the distance between the two nodes is to be elongated, is increased by this one chemical. We call it as a hormone, that is a gibberellin. Second one application is that it break down the dormancy of the seed and the bud. So in case of this one, so in the germination period, some of the plants are there that are germinate only in case of the light. So they are not able to germinate in absence of light. So if you are going to give this one gibberellin, so these seeds or the birds are there that are to be germinate in case of the dark also. Means if there is no light is present, though this seed or the birds are there, they show their germination. Why this is so? Because of this gibberellin it causes the release of this one chemical alpha amylase and other hydrolytic enzyme. So these alpha amylase it causes the breakdown of the starch, or we can say that the polysaccharides. So when this one starch is breakdown is converted or released the maltose as well as the glucose, and that glucose is utilized by the seed or the bud for their growth to break down the dormancy. So in this way, the breakdown of the dormancy is occur. Now the third one function is that it increase the length or the size of the leaves, and because of this one, Giberellin, it shows the number of the leaves formations as well as the increase in the length or the shoots of this dominance. Then it also utilized in case of the induction of the parthenocarpy. Parthenocarpy is nothing but the seedless fruit formation without the fertilization. What are the fruits are there, that is you form, and if the seeds are not there, so such type of the fruits are known as a parthenocarpy. For example, your grapes are there, tomatoes are there, like bees are there. So these are the fruit formation is to be initiated by this one gibberellin. Next one function is there, it delay in the sense of the leaf as well as the citrus fruit, or for example orange, lemon is there, means it delays the aging of the fruits are there. Suppose these are to be utilized if you want to preserve the food fruits for a longer period of the time. So or if you want to transport this one particular fruits to the other area where it requires a time. So in this case, the gibberellin plays an important role that delays the maturity or the ripening of this one fruits are there. So in this way, the fruits remain in an unripe condition for a longer period of the time. Next one is there, it promotes the bolting and the flowering. Long bean plants are there. So wilting is nothing but the initiation of the same formation where the flowers are there that should be occurred. So on this one particular branch, there is a initiation of this one particular branch formation for the flowers are there. So such type of the condition is known as the wilting. And the what the flowers are there that is to be uh, occur only that requires a whole day for the flowering. So in this case, gibberellin that reduces their flowering time, means without a whole day Within a short period of the time or the half day, the flowering is initiated. So likewise, the function is performed by Giberellin. Next one is there, it lower the dose, increase the production of the secondary metabolite. Means in a small dose, when the Gibberellin is to be given, what are the secondary metabolites are there that is to be improved, or the concentration of the metabolite is more in the particular leaves are there or the shoots are there. For example, cardiac glycoside per shoot, digoxin, digitalis are there. In case of this one, digitalis plant, the cardiac glycoside concentration is more where the gibberellin is given. Next one function is that it increases the production of the digoxin in the leaf and the root culture of the digitalis. Suppose by if you are going to isolate the particular cell is there, and you have to be cultivate that one particular cell in a plant tissue culture by utilizing a nutrient agar media, and if in this case if you are going to provide the gibberellin, so in that one culture, callus culture, the concentration of the digoxin is found to be more in the callus where the digoxin, or sorry, gibberellin is given. Next one function is there, it increase the volatile oil concentration. For example, mentha is there, avenos is there. So in this one particular plant, the concentration of the volatile oil is more, where the gibberellin is there is to be provided in a small concentration. It promotes the fruit growth. Then it also increases the maturity or the ripening of the fruit. Sorry, fruit size is there should be increased. It delays the fruit ripening, or we can say that maturity of the fruit. Increase the size of the fruit.